He'll be leading us through a fascinating discussion this morning, and I'm now going to turn it over to Jake.
I've got this one. Great. Thanks. It's a pleasure to be here. So I'm going to talk to you about the future of biofuels as we see them. I lead an institute that's funded by the U.S. Department of Energy, where we work on all aspects of producing biofuels from biomass. But before I get started, I thought we'd talk a little bit about the problem. Now, I have a few slides here to go through, and I'll walk through those. If you want any discussion or have any questions along the way, please don't hesitate to interrupt me. So let's see if we can get the first. There we go. So let's just talk a little bit about petroleum and where we stand today. So we use roughly 225 billion gallons of transportation fuels annually. So that's in the form of gasoline, diesel, jet fuel, and the like. We also use a huge amount of petroleum for producing chemicals. In fact, nearly everything that we're in contact today has some portion of petroleum in it. The carpet fiber here is made almost exclusively of petroleum. The plastic on this podium, the plastic on the chairs, some of the materials, the ceiling that goes into the tiles, the paint, almost everything has been derived in some form from petroleum. So we're hugely dependent on it. If you look at a barrel of petroleum, roughly 15% of that petroleum is used for producing all the chemicals that we want. So it's amazing that 85% of that goes into transportation fuels, and only 15% of it's used to produce a huge number of chemicals. So I thought I'd just give you a partial list of some of the come from petroleum, and this is a pared down list that I got from the internet. It's much more extensive if you go on the internet. It includes things like shampoo, upholstery, movie film, pantyhose, synthetic rubber, and the like. So a huge number of chemicals. It just emphasizes how dependent we are on petroleum as a resource. Now, the challenge for us, of course, is that the petroleum reserves are dwindling. In the U.S., petroleum discoveries peaked in 1972 and have been dwindling since, and production has been dwindling with that. And you can see kind of as you march through different areas of the world where petroleum discoveries have peaked. And if you look at the very top of that, sometime in this decade, we anticipate that worldwide petroleum discoveries will peak and then start to decline. Now, these numbers aren't hard numbers. They're not exact numbers because countries like Saudi Arabia protect the petroleum reserves and the numbers. So we don't actually have good estimates for countries like that. Now, the challenge is that peak discoveries will happen sometime in this decade, and yet we have all of these economies ramping up. China is ramping up. India is ramping up. Their economies are growing at double digits, and they would like to have the same lifestyles that we have in this country. So that's just those two countries alone, half the world's population. So the increase in oil will continue. The demand for oil will continue, and in fact, it will increase with time. And that's going to mean that prices are going to continue to rise. We're going to have, I think, short-term changes where the prices might go down. In fact, they're trying to release oil from the strategic petroleum reserves to drive the price down in the U.S., but those are temporary things. Over the long run, the prices of petroleum will increase dramatically. Now, in 1972, when U.S. petroleum reserves peaked, or discoveries peaked, from that point forward, we started to import oil. Huge amounts of oil, and now about 2/3 of the oil we use is imported. And it's imported from some rather unstable parts of the world, which means that we're sending our cash to those parts of the world as well. Now, if you just look at the amount of energy that's consumed annually, you can see that nearly all of that comes from petroleum in some form or the other. And there's a small fraction that comes from renewables. Roughly 8% of our energy comes from renewables, and of that renewable, 20% is biofuels. So that means that right around 1.5% of the energy we use is in biofuels. So a very, very small fraction. Now, in the U.S., that biofuel is almost exclusively, not entirely, but almost exclusively ethanol, and ethanol that's produced from corn. So corn is a great source for sugar to produce something like ethanol. The corn has the sugar all wrapped up in a very convenient location in the ear of corn, and in fact in the kernel, and you have starch that's very easy to break down that's in that ear of corn. And when the plants grow, they consume carbon dioxide in the atmosphere, so it scrubs the atmosphere of carbon dioxide. We, of course, pick the corn. We farm it, incidentally, just to state all conflicts of interest. I'm from a corn farm in Nebraska, and so I know this firsthand. That corn is picked, it goes into a biorefinery, a fermentation facility, where the starch is extracted, broken down with enzymes, and then yeast ferment that into ethanol. Now, if you look at ethanol production over the last decade or so, it's increased tremendously. And fortunately, it's not for beverage consumption. It's for fuel consumption. And if you look worldwide, ethanol production has increased just dramatically. A lot of this has been in the U.S. and in Brazil. Now, I should mention, I just came back from a trip to Brazil where they're producing their ethanol from sugarcane, and it's an extremely efficient process. As I mentioned, when we produce corn this way, we scrub the atmosphere of carbon dioxide. If you burn the ethanol in your car, you're putting that carbon dioxide back into the atmosphere. So we have the chance of having a fuel that's carbon neutral. But unfortunately, there are many challenges with using corn as a feedstock for ethanol. The first challenge is really around the water requirements. Corn takes a huge amount of water. If you compare that to, say, sugarcane in Brazil, they use nearly no water. It's all rainwater, so the water is essentially free. We also use a huge amount of fertilizer. Roughly 1/3 of the energy used in agriculture goes to produce nitrogen-based fertilizers, and these are used almost exclusively for fertilizing corn. 1% of the world's energy goes to nitrogen-based fertilizers, so a huge amount. If you look at the energy balance on ethanol from corn, if you consider all of the energy inputs, the diesel that you have to put into the tractors, to plant the corn, to farm, to cultivate the corn, to pick the corn, and then to turn it into a fuel, the energy is about even. That means that you put in 1 unit of energy and you get out about 1 unit of energy. There have been calculations that say you even get out less energy than what you put in to corn. So there are a huge number of challenges. If you compare that to the energy inputs and outputs for ethanol from sugarcane, they get out about 7 to 8 times the energy that they put into the sugarcane. That's because, 1, they don't have to fertilize it. 2, you only have to plant once every 7 years. So you get essentially 7 cuttings out of sugarcane, and you only get 1 cutting out of corn. So a large number of challenges in using corn as a feedstock for ethanol. Now, we'd like to use other types of biomass, and there are some convenient forms of biomass, like straw, like tall grasses. This guy here is standing by a tall grass called Miscanthus giganticus. The name is pretty obvious. It grows to somewhere about 10 to 12 feet tall. It's a native Chinese grass, but they've been cultivating it at the University of Illinois for about the last 20 years. It uses much less water than corn. It takes almost no fertilizer, and you can get many cuttings off of it. So it's very similar to sugarcane, except it doesn't have sucrose. It has cellulose, as I'll tell you about in just a minute. Now, why should we use biomass? Well, as it turns out, we have a lot of it. If you just look at the amount of biomass that goes unused on an annual basis, it's somewhere on the order of 1 billion tons in the U.S. alone. The U.S. Department of Energy and the U.S. Department of Agriculture came up with this number several years ago, and it's in the form of corn stover, so that's everything but the kernel of corn, straw, perennial crops, so these are things like grasses like switchgrass, miscanthus, forests, so there's a lot of trees that fall down in the forest that could be used. There's some grains that might be better for fuel use or could be used for fuel use, and other crop residues. So a large number of biomass. Unfortunately for us, that biomass isn't easy to get access to. But if we could get access to it, here's what we could do with it in theory. So if we just compare that 1.3 billion tons of biomass with the amount of energy or petroleum used in 2007, we used about 7.5 billion barrels of petroleum in 2007. Roughly 2/3 of that was imported, and 1/3 was domestically produced. Now, those numbers don't quite add up because there are some gains and losses in the refinery. Remember, we make things like tar from petroleum and a few other things. Now, if we compare that 1.3 billion tons of biomass, if we just burned that and compared that to the energy in a barrel of oil, you'd come up with something just shy of U.S. imported oil. So that's a billion tons of biomass burning. You get almost the energy if you burned that imported oil. And if you converted this into biofuels, there's going to be some inefficiencies because you can't use all of the plant to produce a biofuel. You can get almost to domestic production of petroleum. So the goal going out for the government, and this seems to be somewhat of a consensus, is that in the next 20 years, we should be able to replace about 1/3 of the petroleum we use with fuel produced from biomass. 1/3. Now, here's the challenge. As I told you, corn in many ways is an ideal crop for producing fuels. It's not very efficient. It takes a lot of fertilizer and water, but it has all of the resources, the sugar that you want to convert into the fuel, stored in that kernel of corn. And it's very easy to pick that off of the plant. Unfortunately, biomass is much more complicated. So this is a plant cell wall, or what would be left of a plant cell wall. Those holes in the middle are where plant cells lived. And after the plant cells have died out, and you've cut the biomass, this is what's left. Basically, those cell walls, those outlines of where the plant cell was. And those cell walls are pretty complicated. And I've got an artist rendition here, and I'm going to describe what's in that cell wall. Basically, plant cell walls, plants in general, are made of sugar. It's a complicated sugar, but they're made of sugar. There's 2 types of sugar. We'll call it hemicellulose and cellulose. Cellulose are great sugars. These are the same kinds of sugars that we might find in sucrose and other sources of sugar. They're basically glucose. Hemicellulose, they're okay sugars. They're not the best sugars, but they're still pretty good. Lignin, on the other hand, is not great. It can't be used as a source for biofuels right now. It's a polymer that wraps the cellulose and hemicellulose. Recall that there are lots of critters out in the environment, beetles, fungi, mushrooms that want to grow on the plant and use their sugars. And so this lignin wraps the cellulose and the hemicellulose and protects those sugars so that all the pests out in the environment don't eat the plant alive. Now, that cellulose, that good sugar, is pretty complicated in and of itself. So cellulose is what's cotton is made of. So this shirt I'm wearing is 100% sugar. It's cellulose. Now, if you think about this sugar, I launder this, or somebody launders it for me, but they throw it in water, and it doesn't dissolve and melt away. So that tells you something about those sugars, that they are really strong. They're basically linked together in what we call polymers, so multiple sugars linked together, and they're very strong. So if you want to get the sugar out of this shirt, you have to add enzymes. Enzymes are biological molecules that break down different components. So you have enzymes in your laundry detergent. They take the stains out of your shirts or your clothing when you get stains on them. They also prevent all the fraying on the shirts. So in the old days, before they added enzymes, you'd get fuzz balls all over where the ends of the cotton started fraying. You don't get that as much now because they add enzymes that clip those fuzz balls off. We have to add very strong enzymes if we want to get access to those sugars and use them to produce fuels. So there's a couple of processes that you have to use in the modern biorefinery. The first is a pretreatment process. This extracts that lignin, that tough polymer, away and gives you essentially the sugar-containing polymers, cellulose and hemicellulose. And then we add those enzymes that break down those strong polymers of sugar and turn them into sugars, single units of sugars that can be used by microbes, yeast in our case, to produce ethanol. So that's the entire process one could use if you wanted to produce ethanol from cellulosic biomass. Now, there are some challenges, and I'm going to talk about the science we're doing to address some of these challenges. We'd really like more good sugars, more cellulose. We'd like less of the sugars that aren't quite so great, the hemicellulose, and we definitely like much less of the lignin. Now, there have been studies to remove the lignin and the plants die, so you can't remove it altogether, but maybe we could have less lignin, or we could have it in certain places in the plant so that it's more convenient to get the sugars, the good sugars, out of the cellulose. We'd also like to have some better fuels. So if you think about it, we're using yeast to take sugar and produce ethanol. Ethanol is what nature gave us. It's okay as an oxygenate in gasoline, so if you own a standard gasoline car, you can drive it on about 10% ethanol. I hear in Brazil now that they're putting up to 25% ethanol into gasoline, but it's against the law here in the U.S. The automobile manufacturers are fighting that, in part because they don't know the impact on the engines. So we've got this limitation in how much ethanol you can put in gasoline. If you have a flex fuel car, you can get E85, which is 85% ethanol. Now, when I talk to my colleagues at Exxon, they say, oh yeah, it's great. People have these flex fuel cars. They normally are getting gasoline every Saturday, filling up for the week ahead. And the first time they put in E85 into their car, they find that they can't get to the next Saturday, that they've got to fill it up on Thursday. And it's the last tank of E85 they buy, even though they've got a flex fuel car. And the reason is because ethanol has less energy in it than gasoline. So a gallon of ethanol will get you about 2/3 of the miles that a gallon of gasoline will get you. So people don't want to buy it. What's more, we can't transport ethanol through pipelines. It's corrosive. So we have to use rail cars or trucks to transport ethanol. And finally, it's poisonous to the yeast. So we only get about 20% ethanol in a fermentation, and we have to distill the ethanol out to purify it. I like to say that ethanol is better for drinking than for driving. So if you wanted a fuel for your transportation infrastructure, you'd like a fuel that behaves identically to the fuels that we're now using as gasoline, diesel, and jet fuels. So we want advanced hydrocarbons, identical to the hydrocarbons we get from petroleum. So I'm going to talk about each one of these problems in succession. And the first problem I'm going to talk about is developing better plants that have more of the good sugars, less of the bad sugars, and much less lignin. So there are genes that control expression of all the genes in the plants. They're called transcription factors. They're master regulators of the plant cell. And at the Joint Bioenergy Institute, one of our scientists has identified a key transcription factor that regulates how much of the good sugars and how much of the bad sugars and lignin that a plant produces. And by changing how those transcription factors, those regulators, are expressed, he's been able to change the amount of good sugar and bad sugar and lignin in the plant. And that's shown here, just diagrammatically. So on the left is the control plant, the plant we started with. And this has got the standard regulation in the plant. On the right is where he's changed the regulation of the sugar production in the plant. And you can see that the plant cell walls are much thicker. And I've shown that just diagrammatically here. So again, in the normal plants, you have a fairly thin plant cell wall. And the rest of the plant cell is used for, in many cases, carrying liquid up and nutrients from the roots of the plant to the top of the plant. But it has many more of those channels than what it needs. And so what he found is that by manipulating these transcription factors, in fact, you could fill up some of those vesicles, some of those vessels that carry the nutrients from the roots of the plant to the top of the plant, and fill them up with cellulose. So what this is like is getting more fuel out of your biomass, or making a much denser biomass. It's got more sugar in it, and therefore we can produce the same amount of fuel from much less biomass. One of the key issues in the future is going to be transporting all of that biomass to a biorefinery. This means that you have to transport much less of the biomass. Now, the other challenge is this lignin polymer that wraps the cellulose in the hemicellulose. And this is just a picture of the structure of that lignin polymer. It's very complicated. It's very difficult to break down. In fact, what we do with lignin right now is we burn it and we turn it into electricity, or we burn it and use it to distill the ethanol from the fermentation broth. What we'd really like to do is make cuts in that lignin, make it less polymerized, less extensively interlinked, so that it would be easier to break down. And so researchers at J-Bay have come up with a method to introduce genes into the plant cell that allow you to make a lignin that's more easily broken down, and you can get better sugars out. So here's a time course of sugar released with time. You can see on the left are the wild-type plant, the plant we started with, and at the bottom is the engineered plant. When you use this engineered plant that has this degradable lignin in it, we can release much more sugar over time. That means that you can get a much greater amount of biofuel from the same amount of biomass. So these two features together give us a much better plant that we can use to produce biofuels. Now, in the last part of my talk, I want to talk about this last step of engineering microbes to produce advanced fuels. As I mentioned, we only have ethanol available to us right now as a biofuel, and it's really what nature gave us. But we can't use it in jet engines at all. We can't use it in diesel engines. It can only be used in gasoline engines. So we'd really like to be able to produce gasoline, diesel, and jet fuel replacements. So the challenge is we need to engineer a microbe that can produce these kinds of fuels. And I'm going to talk about how we're going to do that, but realize that if we can engineer a microbe that can produce these kinds of fuels, we can also engineer a microbe that will produce chemicals. But how are we going to do that? How are we going to engineer the next generation of biorefinery inside a microbe? Well, let's consider for a minute how we produce a chemical like styrene. So styrene is one of the most widely used commodity chemicals. It's produced from petroleum in a petroleum refinery. And we build that refinery by pulling together devices to construct the chemical refinery. We bring in distillation columns and reactors. They all function independently. And we link them together using standard connections, pipes. Now, this way of building a chemical factory is very convenient because it means that a chemical engineer, I'm a chemical engineer by training, can pull together all of these different, what we call unit operations, build a chemical factory very quickly, and switch from producing styrene to producing rubber to producing plastics very easily. And this has allowed the chemical industry to produce all of these products that we now get from petroleum. Well, we'd like to do the same thing with a microbe, engineer it in the same way that we build a chemical factory. Now, the proof that we can do this is actually one of the projects that I worked on early on, and that is to engineer a microbe to produce a different kind of hydrocarbon, in this case a hydrocarbon that's the best, most widely used anti-malarial drug called artemisinin. Now, just a bit about this hydrocarbon. It comes from this plant, Artemisia annua. It's a cure for malaria, but it's in extremely short supply. Right now, there is a real need for artemisinin. It was overproduced in around 2005. There was an oversupply in 2007. But all of that stockpiled artemisinin is now gone. And if you look at the projections for the demand going out to mid-century, they mean there's going to be a lot less artemisinin produced. The only way that we're going to make up for that is by increasing the cost, and the people who get this artemisinin, who need it, are people who only earn a very small amount of money, less than $1 per day. So my lab engineered a microbial factory to actually produce this drug by cobbling together genes from the plant that naturally produces it and from different microbes. Now, the way we do this is not like how we engineer a chemical factory. We go out and sequence plants, we get the genes from them, and this gives us a natural parts list that we can then use to build the chemical factory. These parts aren't standardized. They're completely unstandardized. So when we put them together, it's very challenging to get them into the microbe and to get them to work. As I said, there's no models for how to put these together. When we build a chemical factory, we have models for how they work. We have standards for construction, but we have none of that. And so when you build a microbial chemical factory, we often run into problems where things just don't work, where you might accumulate some toxic chemical inside the cell that kills the cell. So that project alone took us about 6 years. Over that time, we increased the production by about 10 million fold. But it took $25 million and 150 person years to actually complete the work. Fortunate for us, we had funding from the Bill and Melinda Gates Foundation. We don't want to spend that amount of time and that amount of money engineering microbes to produce advanced biofuels, because we're going to need a huge amount of biofuels. So that's the whole idea behind synthetic biology, is that we can reduce the time and the cost to engineer biology by having well-characterized components that we can assemble together to build microbes that will work better. So we need genes and genetic controllers that have standard connections, much like pipes have standard connections. We need these parts to be characterized. So this is an example of a data sheet for a part, much like you would have a data sheet for a pipe that you might buy that you're going to assemble together. And we have to have robotics and standard connections in order to put all these genes together and make the controllers. And on top of that, we need computer-aided design in order to assemble all these components. Well, why should we be doing this now? What's changed about biology over the last few years? They say this is going to be the century of biology, but why is that? Well, I just want to compare it to changes in another industry, microelectronics. So this is Moore's Law. So for those of you who don't know about Moore's Law, it was developed by Gordon Moore, actually right around 1968, 1970. And basically Gordon Moore, founder of Intel, predicted that every 18 months, the amount of information that you could pack on a chip would double. And if you look at Moore's Law, so he predicted that, let's say 1970, it's held true throughout. Now, everybody's worried that we're going to fall off Moore's Law, but that line's pretty straight. Pretty amazing prediction over a 40-year period. And that means that basically our computers are doubling in terms of speed or processing power every 18 months. Now, if we compare that to biology, that's the growth of DNA sequencing. In fact, I extrapolated from about 1990 to present, but it's really growing much faster than that. So the amount of information that we can read in biology is growing even faster than Moore's Law. And the amount of information that we can write, so synthesizing DNA, is growing just as fast. And what that means is that the costs are coming down. So sequencing costs are dropping dramatically, and so are DNA synthesis costs. And because sequencing costs are dropping so dramatically, you can now submit your own genome and have it resequenced. And in fact, they're sequencing bits of genomes now, and very shortly, for probably $1,000, you're going to have your full, complete genome sequenced. But what that means for engineering biology is that we can do it much faster, much easier, much better. So how will this help us make biofuels better? Well, we want to engineer this microbe that will produce all these different kinds of biofuels. And luckily for us, we had started with a microbe that produced a hydrocarbon. In this case, a hydrocarbon that's used as an anti-malarial drug. Well, that anti-malarial drug is very similar to diesel fuel. Stephen Colbert joked with me that we were trying to sell diesel fuel to poor children in Africa. It's not the case, but what that means is that we can take that microbe that we engineered to produce the anti-malarial drug, swap out a few genes that encode the anti-malarial drug, and put in different genes that will allow us to produce things that will work very well as advanced biofuels. And we're currently doing this. We've engineered microbes that will produce these fuels. And very shortly, there are a few companies that are now commercializing these. Amyris and LS9, both were developed out of the work from my laboratory, and they're commercializing some of these fuels. Now, the great thing about these fuels is that they're like oil and water. The microbe actually secretes the fuels. They flow out of the microbe and into the fermentation broth. So on the left is a tube. This is a culture of E. coli that's been engineered to produce fuels. And you can see there's an oil layer on the top. That's the fuel that actually separates on the top. And on the right is a micrograph. So those little elongated dots in there are the E. coli swimming around. And those blebs that you see are the diesel that they've actually produced and have secreted. And that diesel can be gotten very easily and purified